Hello students, this is Dr. Faizan Mirza. We are discussing protein mediated transport. Protein mediated transport basically tells us that whenever a substance needs to cross the cell membrane, at times there are substances which need a protein in the membrane for transportation to occur. These proteins are intrinsic proteins. Intrinsic proteins means that they actually are transmembrane protein. An intrinsic protein is one which is present in the bilayer, and a transmembrane protein is an intrinsic protein which is spanning the entire width of the bilayer, which means it's crossing both the monolayers, and it will have one part extending to the ECF surface and the other part extending to the ICF surface. So transmembrane proteins, they work as transport proteins in mediated transport. They could either be channel proteins or they could be carrier proteins. So this is generally called as mediated transport. The mediated transport could either be facilitated diffusion or it could be active transport. Both uses transport proteins. Active transport can then be either primary or secondary active transport. Facilitated diffusion, since it's diffusion, it equilibrates the substance moving in either direction, because since it's diffusion, it's actually just down the gradient, and it's just using the kinetic energy of the molecules. We use examples of uniports here, or symports, or and antiports. What's a uniport? You can see that this is a phospholipid bilayer. This is facing ECF and this region is ICF. And in facilitated diffusion, there will be proteins in the membrane which are transmembrane proteins that can be working as uniports. A uniport is a transmembrane protein that allow movement of one substance at one time in one direction. Active transport, as you know, is against the concentration gradient. So primary active transport is against the concentration gradient. It makes use of ATP breakdown. And secondary active transport is against the concentration gradient as well, but down the electrochemical gradient. The secondary active transports are the ones where symports actually occur, and antiport also occur here. What are symports and antiports? Symports are co-transporters which move substances more than one in one direction, and antiports are counterport transporter proteins which move more than one substance in different directions at the same time. For example, this phospholipid bilayer is of a cell, and this is a cell membrane. I'm adding just more phospholipid molecules to it. And apart from these uniport proteins, let me add some more proteins here, which again are transmembrane proteins, and these transmembrane proteins could be used in moving the substances into and out of the cell. For example, these both are allowing coupled transport to occur. What do you mean by coupled transport? Coupled transport means that the movement is coming from the movement of two substances either in the same direction if it's co-transport, or in the opposite direction if it's counter transport, and it is the binding of the substances at the ends of this protein that triggers the coupled transport to occur. If the coupled transport is using energy from the ATP breakdown, then that means that the coupled transport is active transport. And if the coupled transport is not using the energy from ATP breakdown, so it could be secondary active transport. What are the properties of mediated transport, which means transport occurring through the membrane proteins? Initially, the transport rate is very high because the movement is occurring at a very fast rate, but soon a transport maximum Tm is reached, where further increase in the concentration will not increase the rate of transport further. So if you draw a graph of increasing concentration on the x-axis and the rate of transport on the y-axis, we see that the graph is very steep initially, showing a very high rate of uptake initially, but then a TM or transport maximum is reached, where increasing the concentration of that substance around the cell would mean that the diffusion gradient is very steep. But since all the mediating transport protein channels or carriers are occupied, so irrespective of how much you increase the concentration, the rate of transport uptake cannot go beyond this. The only way to increase the rate of uptake beyond this is to have even more number of channels and proteins on that membrane. But again, they will become constant once the increasing concentration will saturate all of those. This is called as saturation kinetics. Another important feature of membrane transport protein through mediated transport is that it is way more rapid for a molecule of same molecular mass and lipid solubility, which might be moving by simple diffusion. But then soon saturation kinetics is achieved, which means transport maximum is reached, and increasing the substrate concentration will not increase the rate of reaction. If you just try to align it with simple diffusion, we see that if simple diffusion you increase the concentration of the substance, the rate of transport keeps on increasing linearly. So if this was simple diffusion, the graph would have just gone straight linear without becoming parallel to the x-axis. The mediating transport proteins are highly chemical specific as well as stereospecific. Chemical specificity means that each carrier protein or a channel protein will be specific for a certain solute and not all. And these are stereospecific as well. Stereospecific means that there are stereoisomers that can rotate light into a different direction. So stereoisomers also do not move by the same protein, transmembrane protein. For example, dextroglucose and levoglucose, they both are two stereoisomers of C6H12O6 glucose molecule. They both have generally the same chemical formula, but they are stereoisomers. So should they be going through the same transport protein? One expects that since they have the same molecular formula and the same chemical formula, so they should go by the same protein. But since they are isomers, one can rotate light in the right direction and the other can rotate light in the left direction, dextro and levo, so they do not use the same transport protein, even they use different transport proteins. So this is the level of specificity that you can expect in a particular transport proteins. The way you have studied competitive inhibition and non-competitive inhibition in enzymes, the very same this concept aligns here as well, that competitive inhibitors, which are similar structure, they can compete for the same protein, and non-competitive inhibition can also occur where a structure which is not exactly similar to the one which is entering through the protein will still bind to it. For example, there are drugs, for example, there are toxins and venoms, for example, nicotine, that's a drug, and toxin muscarine, or venom of cobra venom. All these have specific target sites where they go and they bind to a specific protein present on the membrane of the cell, thereby disrupting the normal functioning of that protein and affecting the overall functioning of the cell. That's all I'll be covering up in this video. Thank you so much.